Hello, you are most welcome, ladies and gentlemen, to Real Impartation Moment on Tuesday Night Anatomy with Daniel Open. Today's section, we continue with the nervous tissue that we've already begun. It means that there's going to be the second part of the series, okay? So it means that if you've not watched the previous first part video, kindly get the video link and watch it. Of course, last week we didn't have a meeting. Last 2 weeks, yes, I was celebrating my 3 years of being on YouTube. Yes, for that reason, we went on an intermission. Now we've resumed this kind of Tuesday night anatomy with Daniel Open. So make sure that any moment, any time, even after vacation, you'll be seeing me. Yes, so that I will explain all these concepts for you before you come back to school. And so, without much ado, let's look at what we have to do today. Today, we want to tackle something on the neuroglia. Yes, the glial cells that we began looking at. We looked at it briefly, especially we talked about the oligodendrocyte. We talked about the Schwann cells doing myelination for the CNS and the PNS, neuronal axons, respectively. We looked at those ones. We are going to look at other details of these guys. Okay, we are going to look at them. And then finally, I also talk about the types of synapses by way of the chemical synapse, okay, which is an evolutionary improvement of what we call the chemical synapse. We will see that. So I believe you are going to be with me so that we run through this one. Yes, after that later, we'll look at the other things. So today is going to be a brief, thing that we are going to do today. So we're going to look at what we call neuroglia. Neuroglia. Yes. Now, anytime we talk about the neuroglia, yes, I told you that the neuroglia, in terms of population, they are way more than that of the neurons, which are the excitable cells of the nervous system. Yes. In fact, I told you that at least they are 10 times more than that of what we call the neurons, at least. So one thing we see is that these neuroglia, something we also call glial cells. So if you don't see neuroglia, you may see glial cells. So neuroglia, otherwise something we want to call glial cells. Yes. So these glial cells, we have some in what we call the CNS. Then, my friends, we also have some in, of course, the PNS. Okay. So we have some in what we call the central nervous system, CNS. And then we also have some in, of course, the PNS, peripheral nervous system. Peripheral nervous system, yes. So that's what we have. Now, in the central nervous system, we have 4 of these neuroglia. These cells, which we are calling them glial cells, we have 4 of them. Okay, so in the central nervous system, we have 4 of them. And who are these guys we're talking about? We have something we call astrocyte. Astrocyte. Okay, so first, let us get to know their names. Astrocyte. Then, we also have Ependymal. Ependymal cells are also present, yes. We also have what we call the microglia. Microglia. And then finally, we also have what we call the oligodendrocyte, the one we've seen already, oligodendrocyte, or you can say oligodendrocyte. Yes, so these are the 4 glial cells we have in the central nervous system. Now, how about in the peripheral nervous system? In the peripheral nervous system, we have just 2. Yes, we have 2 of these glial cells in the peripheral nervous system. So in the case of peripheral nervous, we are going to just talk about 2 of them. And who are these 2 guys we are talking about? Simply, one of them is called satellite. Satellite cells are present over here. And then, of course, we also have what we call Schwann cells, the one we've seen already. Okay, now, the first thing, if you look at the astrocyte, the word astro, has to do with the star. So, astrocyte, a cell that resembles the star. Okay, so, for instance, it may be something like this. Let me guess. And it may be having all these processes around. Okay, making a stellate. An oval shaped cell having all these processes. So this is the astrocyte. Now, what is this astrocyte we are talking about? Whenever we talk about this astrocyte, the first important thing that I want you to be aware is that it maintains the blood-brain barrier. Yes. This astrocyte over there will bear some food processes. I'm going to explain all those ones for you. It will bear some kind of food processes. The astrocyte will bear food processes. And these food processes, although they are not directly involved in the formation of the blood-brain barrier, it is important in helping to maintain the blood-brain barrier. Now, what do I mean by this? Just a quick one. Just look at what I'm going to demonstrate for you. You may have, okay, now, let's say this is a blood vessel. Now, you are aware that blood vessels, just like lymphatic vessels, will be lined by an epithelial lining, which is simple squamous epithelium, And that simple squamous epithelium over there is the one we simply call endothelium. For this reason, I'm having this endothelium over here. But the point over here is that the endothelium, this kind of blood vessel, the cells over there, by way of the endothelial cells, they are joined tightly by something we call tight junctions. Zona occludens. I'm sure you are aware about these things. Epithelial cells, they mainly, really make use of these tight junctions. These junctional complexes, including the tight junction. So these endothelial cells over here, which are lining the wall of the capillary, okay, the blood vessel in there, in the brain, we are calling it, okay, these guys over here, these are what we call the tight junctions, tight. Now, my friends, keep an eye on where we have this tight junction regarding the blood-brain barrier. I'm talking about the blood-brain barrier, BBB. Now, but the point is, why do we need the blood-brain barrier? The reason is simple. Now, the cells of the brain, just like the cells of the gonads, by way of the haploid cells, eventually becoming functional, okay, in the case of the male, the testes, the functional, okay, the haploid cells in the spermatogenic series, by way of course, the secondary spermatocytes, the spermatids, the functional spermatozoa, they are haploid. Those cells, they were not exposed to the immune system. So the immune system is not aware of them. In other words, whenever any products, okay, of these cells get into the blood, or the blood meets these cells, okay, the immune system, which of course, by way of the antibodies and all that in the blood, will begin to mount an immune response. Yes, because they will see these guys as foreigners to the body. the same way the body fights infections, okay, pathogens that come from elsewhere because they've not been, exposed to the immune system. So the same way the cells of the brain, okay, they don't need to get in contact with the blood. Okay, their product doesn't need to leak in here. Otherwise, there's going to be an immune response against it, an autoimmune destruction of the brain. You don't want that one to happen. And that is why God, in his own wisdom, had to put something in there which are called the blood-brain barrier. Now remember, the blood-brain barrier is formed from the tight junctions joining the endothelial cells of the capillaries in the brain. Yes, at the level of the brain. That's what we see. Now, one thing that you will also be aware is that whenever you have an epithelial lining will sit on something called basement membrane. So there's going to be something which you call basement membrane. So let me say there's going to be a basal lamina of the basement membrane over here. So this black thing I'm trying to show over here, I'm having the basement membrane or the basal lamina of the blood vessel in here. It is also in there already, yes? But then one more thing that happens is that these astrocytes, their food processes will give some kind of support, reinforce this kind of blood-brain barrier that we formed over here. Okay, so that reinforcement. Okay, so take it that there's one, astrocyte, this process is coming here, other ones will be meeting. So it will be something like this. Okay, so there will be all these processes. The food processes of the astrocytes will be like giving some kind of support, mechanical support, and then provide some kind of structural integrity to this blood-brain barrier. In other words, the astrocytes do not form the blood-brain barrier. They help maintain the blood-brain barrier. Yes. That is one important thing that I want you to be aware of. Now, what is this blood-brain barrier doing? We will see another cell type, which I will come to it, the ependymal cells. Now, ependymal cells, they line something called ventricles of the brain, where CSF flows. Also, in the central canal of the spinal cord, I will come to these things, CSF will be flowing over there. So, lining the central canal of the spinal cord, we also have ependymal cells. Yes. Now, these ependymal cells, you may have some over here. I'll come to it again. Yes, I'll come to these ependymal cells again. But for now, you will see that this is the blood. We have the brain tissue all around over here, including what we call the ependymal cells. Now, these are some kind of, okay, let me say these cells. Okay, I'll come to it, I'll draw them again. These are ependymal cells. Now remember that at this level, the blood product, okay, is not getting anywhere to the brain. Through the tight junction, they cannot move anywhere. They cannot also receive any product from the brain. Neither, CSF will be flowing in this manner. CSF can come this way, can flow out, okay, in that manner. But still, that CSF will not get into contact with, of course, the blood. So it means that this blood-brain barrier is a morphological, it's an anatomical barrier, okay, that delimits what we call the blood, okay, from, of course, the brain, including the brain tissue, including, of course, the CSF. Yes, that is what we're going to see. Now, in terms of permeability, I'll be mentioning something else to you, which is something we call blood CSF barrier. And now I'll be coming to it pretty soon. In terms of permeability, the blood-brain barrier is relatively more permeable compared to that of blood CSF barrier. so that molecules which want to cross the blood-brain barrier should be lipid soluble. That's one point. Or they have to be transported through some kind of passive, means, to be carried, to transport across these areas. We will see that. Okay, so that's one thing. The astrocytes, they help maintain blood-brain barrier, number one. Number two, if they are maintaining blood-brain barrier, then it means that they are providing some kind of mechanical support. That's number two. Now, number three, what they are going to do is that they are going to regulate the concentration of ions. Yes, number one, concentration of ions. Not only that, concentration of nutrients. Yes, around over here. Not only that, even some gases, and most importantly, neurotransmitters. Yes, they are going to regulate their concentrations around in the brain. That's number one. The other point. Now, the next thing too that we will see is that now these astrocytes will be relevant also in something we call absorption of what we call neurotransmitters. Absorption of neurotransmitters. Now, why are we absorbing them? The neurotransmitter which has been used will have to be reabsorbed, and these same astrocytes will recycle these neurotransmitters. Yes. So at the level of the cell, by way of the astrocyte, we see recycling. And that's why human beings, we've learned some of these things. Recycling is important over here. But my friends, these same astrocytes will also be relevant in forming something called scar tissue after injury. Okay, so that is what, these astrocytes, they are doing. Okay, maintaining blood-brain barrier, importantly. All right. But just to tell you, although I'm saying that blood-brain barrier is formed yes, by the tight junction, the zona occludens, okay, between these epithelial cells, these endothelial cells of the capillaries in the brain. Yes, in situations they want to ask for more, then you talk about the basal lamina or the basement membrane, including the foot processes of the astrocyte, although they are not mainly contributing to it. Okay, that's an important thing. Good. So that is the astrocytes. Now the other cell we want to look at, review its function, is what we call the ependymal cell. Now for the ependymal cell, okay, let me just show you how it looks like. I drew some for you, but this is actually the complete picture of an ependymal cell. Okay? I have two ependymal cells over here. Yes. Now, this time around too, I want you to draw this. We still have some kind of capillaries around. Now, this time around, these capillaries are not glued together by way of tight junctions. No, there are gaps between them. In other words, these capillaries I have over here, they are fenestrated. They have chemistry. They are fenestrated. So it means some molecules can move out, can come in, move out, come in, okay, in that manner. So what sort of capillary do I have here? Fenestrated. Fenestrated capillary. That's what I have here. Now, in the case of blood-brain barrier, I told you that they were continuous capillaries, which were glued together by tight junctions. This time around, we have fenestrine in the capillary endothelium. Yes, it may sit, yes, so obviously sit on the basement membrane. Yes, basement membrane will be there. Basal lamina. And what do we see? I see these cells. These are the ependymal cells I'm talking about. So first, the ependymal cells, now within the brain, let me tell you something. Within the brain, we have ventricles. Now we don't have ventricles only in the heart, no. We have ventricles in the brain. We have 4 of these ventricles. Now in the cerebrum, later I'll explain all these things for you from the embryonic point of view, explain all these things, okay, so that you understand whatever we are talking about. In the brain, the cerebrum, for instance, we have 2 in this area, left and right hemispheres. You'll be seeing what we call left and right lateral ventricles. Okay, so there will be one here, they will be connected, yes. And then we go to what we call, between something called the thalamus and the hypothalamus, you see another ventricle, which we call the third ventricle. Okay, and then of course, in the region of the brainstem, okay, and whenever we talk about brainstem, we're talking about the midbrain, but not really in the midbrain. Midbrain will have something called aqueduct or sylvius, later we explain. Actually, in the pons and medulla oblongata, and then also the cerebellum, in that region, we also have another ventricle, which is called a fourth ventricle. Again, if you look at the spinal cord will bear a canal, and that canal is called central canal of the spinal cord. Now, over there, what we see is that lining these chambers, we have these cells over there, which we call them ependymal cells. Ependymal cells, yes. That is one thing we are going to see over here. Now, the usefulness over here is that the ependymal cells, together with these blood vessels around over here, will form something we call choroid plexus. Choroid plexus, and that's an important concept I have to explain. Now, so if I have a blood vessel, if I'm having blood vessel, what it tells me is that now this is epithelial in origin. Ependymal cells are epithelial in origin. And then blood vessels, yeah, they are avascular. These epithelial cells are avascular. So necessary, we are supposed to have some kind of connective tissue around where blood vessels will be in there. So that area, okay, that whole thing is called choroid plexus. Choroid. Partly contributed by, of course, these ependymal cells and, of course, these neighboring blood vessels in there. Yes. So that is the first thing I want you to be aware of. Okay, choroid plexus is in there, and then the important cells, epithelial cells over there, they are the what we call the ependymal cells. Now, if you can clearly see over here, the ependymal cells at the apical region, they bear these kind of cytoplasmic processes. Yes. And what are these cytoplasmic processes? These are not cilia, neither are they cilia. These guys, they are microvillous processes. Microvilli. Okay, so they will greatly increase the surface area, this time for secretion. Yes, I'm going to tell you what they are going to secrete. Now remember, because they line these ventricles, the central canal, and run over there will be CSF, it means that these ependymal cells will be relevant in secretion of what we call the CSF. They will contribute to the secretion of CSF. Now to tell you something, CSF is an ultrafiltrate of the blood. Yes. So it's just like the plasma, ultrafiltrate of the blood. Just that it is clear. Expect that it is clear. Okay, an ultrafiltrate of the blood. Now, CSF naturally, It's just having similar character, just like the blood plasma. The only thing is that one important thing is that its protein level is relatively reduced. It's reduced compared to the plasma. But even the protein, majority of the contribution is coming secreted by these ependymal cells. Yes. So there's one thing we see. The CSF will be secreted over here. So this region, I have CSF, cerebrospinal fluid. Cerebrospinal fluid. Now, all these things we are learning about, we will go into the application in the clinical setting. Yes. Why we tap CSF, okay, at what region do we take these things? Okay, cisterna magna, for instance, we take these things. At level of, let's say, L3, L4, we can take it. Okay, lumbar region. Why? Yes, we explain all these things for you. What molecules will be there which indicate that there's an infection, whether it's viral or it's bacterial. We explain all these things for you. So what we are saying is that the ependymal cells, what they are going to do, one, they are going to help contribute to, of course, the production of CSF. That's number one. Number two, if they help to produce it, they are going to help to circulate the CSF so that the CSF can flow. And most importantly, they are going to monitor the CSF by way of the concentration of the ions and all that. It's going to be done by these ependymal cells. Now, one more thing that I want you to be aware is that these ependymal cells, as a cell, because it's very important in secreting the protein, it tells you that it should be endowed with protein synthesis machinery. Yes. And whenever I talk about protein synthesis machinery, I don't need to tell you that we are talking about the rough endoplasmic reticulum. That's number one. Golgi apparatus. Because at the end of the day, the protein that will be made over here is not going to make use of it. It's going to secrete into the CSF. Okay? So for this reason, there's the need for post-modification of the protein that will be made. Yes. And that is why we need Golgi apparatus. And not only that, you know, this thing is energy requiring. For that reason, this cell, ependymal cells, should be well endowed with, of course, mitochondria. So we have numerous mitochondria around over here, okay, generating the energy currency by way of the ATP, so that through hydrolysis, this kind of active process will take place. But there's something that I want you to be aware. There's also another barrier. We've seen blood-brain barrier. There is another barrier. This time around, it's not formed from tight junctions of the endothelial cells in the capillary, no. Unlike the case of blood-brain barrier, no. This time around, it is formed at a junction between adjacent ependymal cells at the level of the choroid plexus. Yes. At the level of the choroid plexus, what we see is that these ependymal cells bearing all these microvillous processes, okay, they are relatively cuboidal, okay, with rounded nuclei. Okay, what we see is that they rather be having the tight junctions. So we have this monolayer of cells by way of the ependymal cells, okay, joined to one another, okay, via these things, tight junctions over there. For this reason, whenever you talk about tight junctions, epithelial tight junctions, they are more towards the apical region. It's not towards the base, neither in the middle, it's more towards the apical region. Okay, so that is what we see over here. Good. So that the blood, okay, from here, when it can leak out, it will not be able to come to the level of the CSF. Neither can the CSF also flow and come in here. In this case, these ependymal cells, the epithelial lining, ependymal cells in the choroid plexus, they are forming what? They are forming what we call helping to form blood CSF barrier. Yes. A functional barrier over here. Yes. A functional barrier over here, which is, very least, okay, relatively having lower, permeability compared to that of the blood-brain barrier. Okay, so that is, one thing that you see over here. Good. That is what we call the ependymal cells. Now, once we are through with this, the next cell type which we want to look at is, of course, the microglia. Microglia, yes. Now, whenever we talk about microglia, we are talking about the macrophage of the brain. I shouldn't even say brain, macrophage of the CNS. Yes, microglia. Microglia. So microglia, just, to show you, it will also be having all these processes, giving some other secondary. So this is the microglia. Macrophage, it therefore tells you that because this macrophage, it will be relevant in removing cellular debris, cell debris, waste, as well as importantly pathogens which may find themselves in the CNS. Yes, and by what means? Via phagocytosis. Phagocytosis, cell eating, a form of endocytosis. That is how these microglia, are going to what? Remove these cell debris, waste, and pathogens. Okay, so that is important thing that I want you to be aware of. Already, we said that we have oligodendrocytes. And in our previous meeting, we said oligodendrocytes are doing the myelination for axons of neurons belonging to the CNS, central nervous system, by way of the brain and the spinal cord. That is what is going to happen over here. So we're talking about the oligodendrocyte. Now the word oligo has to do with fewer, processes. Okay, so one thing that I told you the last time is that oligodendrocytes are very promiscuous. They will myelinate an axon of a neuron at a particular segment, then go to another neuron, myelinate its axon, having all those things. So you may be seeing a cell. Let's say a cell having these processes. One of them will join and myelinate one, whatever here. Another one will join over here and will help myelinate at a point. Okay. Now, if you are seeing a cell like this, we are talking about oligodendrocyte. Now, if you look at the way these oligodendrocytes are doing, just like we mentioned about astrocyte having all these food processes. Okay, so we can liken that kind of food process to these things where the oligodendrocytes are attaching to other, axonal, of axons of other neurons. Then what we say is that these oligodendrocytes may also be relevant in providing mechanical support to these neurons. Yes. Apart from doing the myelination, they will provide some kind of mechanical support. Okay, so that is importantly the cells, the neuroglia of what we call the CNS. Now, the next thing that you need to review is the neuroglia of what we call the peripheral nervous system. Neuroglia of the peripheral nervous system. And already we said that one of them is what we call the satellite cell. Satellite cell. And one thing that I'm always of the view is that satellite cells can be likened to the astrocytes. Yes. Because their function tend to overlap. There's some kind of similarity in terms of their function. Why am I saying that? Because number one, what these astrocytes were doing is that they were actually regulating the ion, the oxygen, the carbon dioxide by way of the gases, okay, and nutrients, including neurotransmitters also. This guy, cell cell, is also doing same in the case of the PNS. Yes. So cell cells importantly will be regulating or will be monitoring concentration of ions and importantly gases, mainly carbon dioxide and of course oxygen in there. Yes, that is one thing. Now, one other thing too that we've seen is that these cell cells, okay, now let me just draw it and show you something. Okay, so this is how the cell cell will just look like. Okay, it resembles that of the, just that the processes are a bit, okay, so let's say I'm having something like this. Okay, so this is the cell cell, just likened to that of what we call the astrocytes. Only that we are not forming blood-brain barrier over here, no. So we're not providing that structural integrity. Okay, so that is one useful thing I want you to be aware over here, the cell cell. Now, in the case of the Schwann cell, Schwann. Let me bring it here. Schwann cell. The key thing is that you see the Schwann cell, I mean, really associated with this kind of myelin. So let's say the sheet is from the cell is in the Schwann cell. Okay, we said Schwann cells, mainly they are going to do myelination for what we call the CNS, the PNS, okay? That's one thing that we see. Of course, the astrocyte will also be relevant in providing that kind of structural support. Because just like, I mean, cell lyses are going to do that just like the astrocyte will do. Okay, so the same way, because this one is forming the myelin sheet, it also gives some kind of structural integrity over there. Okay, so that is, I mean, one thing that we see. But importantly, these cells, okay, Schwann cells, doing the myelination for axons of the PNS will also be relevant in repair of injury. So whenever the neuron, the axon gets some kind of injury, they are involved in that kind of repair over here. All right, so that is what we see for these, I mean, neuroglia. For these neuroglia. Now, so that is the important thing. Now, in future, we will delve more into details of all these things, okay? So that's just an introductory bit that we want to really understand. Now, the last thing that I want to briefly talk about is what we call types of synapses. Types of synapses. Now, another thing too that we have to see, let me just tell you quickly. In the case of the cell cell, the cell cell, unlike in the case of the astrocyte, we said yes, they're also contributing, okay, maintaining this blood-brain barrier. These ones, because they are in the PNS, we said the neuronal cell bodies, collection of cell bodies in the PNS, something we call ganglia. Ganglia. So ganglia over there, they will be covered, okay, so that they provide some kind of structural support, some kind of insulation to them, okay, these by way of these cell cells, okay, that's what they are going to do. They are going to cover all these. That's one other function that they are going to play. So now we want to look at types of synapses. Now, whenever you have one neuron and that neuron meeting, a synapse is formed. The physical structure, okay, between these meeting point of these neurons is a synapse. Okay, yes. Now, the synapse, we have two kinds. The first one that we had, okay, is what we call an electrical synapse. Now, look at this. So we have two kinds. We have electrical synapse, and then of course we have something called chemical synapse. Now the first thing I want you to know is that we move from electrical to chemical. So it means that we should understand what this electrical synapse is and why it was deficient for us to get to the electrical synapse. Now, if this is, let's just take it that this is an axonal terminal of a presynaptic neuron. Now, I'll be explaining to you in our next meeting. We will see all these autonomic neurons, how they move and all that. We explain all those concepts for you. For now, just take it that this is a before we form the synapse, there was a neuron. It's axonal terminal. Okay, so this is a presynaptic neuron. And then below, over here, we also have another neuron waiting for the presynaptic. Yes. So this neuron becomes postsynaptic. Okay? Now, this physical barrier space between them is what we are calling the synapse. So this is the synapse. Now this time I'm talking about the electrical synapse. For the electrical synapse, unlike another synapse we will see, where these, okay, axonal terminal, okay, the bulb over here, the dilated region over here will bear something we call vesicles, which within it will be stored neurotransmitter. No, in the case of electrical synapse, you don't need any neurotransmitter. No neurotransmitter is needed. For this reason, we expect that this presynaptic neuron, the cell, okay, over here, as well as even the post, will be having minimal concentration of mitochondria. Yes, because they are not making any energy requirement, whatever. They are not making neurotransmitters. Neither are they going to have a lot of protein synthesis machinery. Okay, that's one use for them. What we see is that there's a connection. So let me see. What we see is that there's a connection between them by way of a gap junction. So that connection between them, so that molecules can move from here to here rapidly, very rapidly, Okay, so this is what called a gap junction. Now, gap junction, which already I've explained to you, that is otherwise known as communicating junction. Communicating junction. Okay. Now, one thing I want you to be aware is that the impulse can travel from this presynaptic neuron to the postsynaptic neuron, or it will even go vice versa. So it means that it can even travel from here and then come to the opposite, cell, presynaptic neuron. Through ions moving rapidly, the information can go in either direction. Now, what it means is that impulse transmission at the level of an electrical synapse will be bidirectional. Bidirectional, yes. So what we say is that one of these cells, once it gets depolarized, quickly the ions move to the other cell so that it causes depolarization in there. So the presynaptic neuron getting depolarized, invariably, at the same time, instantaneously, the postsynaptic neuron also gets depolarized. Number one. Okay? If this one gets hyperpolarized, this one also gets hyperpolarized. That's what is going to happen. Because they are communicating via these gap junctions, which will work rapidly, very rapid. Although, yes, it's working rapidly, it is least affected by fatigue. We thought that if you are working rapidly, you should easily get fatigued. And I'll explain that to you when I look at the chemical synapse. It doesn't easily get fatigued. And it is also least affected by something we call hypoxia and then pH. Why? Because it doesn't need the oxygen to metabolize anything, generating ATP molecule. No, it doesn't need that. So it is least affected by these kind of things. Good. So that is one important thing that we want to be aware of here. In any case, the distance between the presynaptic neuron and the postsynaptic neuron at the level of the synapse is very small. Just about 0.2 nanometers on average, just about 0.2 nanometers. So from here to here. Yes, that is, what we will see over here. Now, if you look at it critically, I'm saying that they are connected via gap junctions. Already we've told you that whenever we talk about gap junction, the communicating proteins, okay, that channel created by those proteins, we call it what? Connexon. Connexon. Okay, something that we need to be aware of. Connexon. Connexon. Now, if I look at this connexon, whenever I have one connexon is something that everyone knows. We are having, it means that one connexon is formed by 6 connexons. 6 connexons. This time around, it is an I, this is O. The channel created from these 6 connexons will give us a connexon over here. Now look at something. The channel in here, if you look at the way I drew it, you can clearly see that partly this whole entire channel is contributed by, of course, the presynaptic neuron and another by the postsynaptic neuron. For this reason, the connexon, which is the channel transmembrane protein between these kind of presynaptic neuron and then, of course, the postsynaptic neuron, is formed by, of course, hemi-channel or hemiconnexon provided by who? By the presynaptic neuron. And a hemi channel or connexon provided by, of course, the postsynaptic neuron, and together, partly by the presynaptic neuron, partly by the postsynaptic neuron, we form the single connexon. That's what we want you to be aware of. Good. Now, if one cell gets depolarized and the other gets depolarized, one gets hyperpolarized, the other gets hyperpolarized, then we cannot really produce a highly compartmentalized, system where we can control these things. For this reason, evolution, okay, happened so that we evolved from electrical synapse to something called chemical synapse. In the case of chemical synapse, we make use of neurotransmitters. And those neurotransmitters, some of them could be excitatory. So for instance, if the presynaptic neuron is depolarized, the postsynaptic neuron will also become depolarized. Some, however, could be inhibitory. Yes, it may cause inhibition to the postsynaptic neuron, okay, to become hyperpolarized, for instance. Okay, so these are the things that I want you to be aware of. Now, so in the same way, let me just sketch out something for you. Okay, now in the case over in the synapse, physical space can be about 10 to 20 nanometers, a bit wider compared to that of the electrical synapse. Now, one other thing too that we see is that we don't have that kind of transmembrane protein by way of the connection over here, seminal communicating channel, no. By these synaptic bulbs, these dilated regions, they will bear vesicles. And within those vesicles, you have what we call neurotransmitters. Neurotransmitters in there. So through something we call efflux. Okay, so once calcium begins to come in, efflux of calcium ions will cause these vesicles, which will contain the neurotransmitters, to dock. Okay, so they will come and dock. And once they dock over here, they will begin releasing their neurotransmitters. Releasing it into this synaptic cleft over here, this space over here. So that what will happen? The cells at the open side, which becomes the what? The postsynaptic neuron, will bear something we call receptors. Will bear receptors. Okay, and those receptors, okay, will interact with these neurotransmitters. And once there's that kind of interaction, then this one will either, okay, will cause some. Now, there are two main kinds of receptors I want you to know at the level of the chemical synapse. We have something called ionotropic receptors. So for ionotropic receptor, quickly, they open ion-gated channels, okay, for these kind of, impulse transmission take place. So that if this cell is depolarized, this one also get depolarized eventually. But sometimes too, it may open. It may be related to some kind of other receptor, which we call them metabotropic receptors. And those ones usually are coupled with some other proteins, okay, G proteins and what have you. Okay, so that, yes, the process becomes a bit, let me say, it doesn't occur rapidly compared to that of the ionotropic receptors. Okay, that's what we see. Now, because these ones, they are using receptors, the postsynaptic neuron bearing receptors, then it tells us that these receptors can easily get fatigued. Yes, they can get fatigued all the time. They have to bear. They can get fatigued, and this also the one way is also susceptible to pH changes and also oxygen concentration. So when there's hypoxia or all these things may affect the transmission at the level of chemical synapse. Okay, so that is what we see. Yes, chemical synapse, we expect that it should work rapidly. Yes, sometimes, we talk about nervous system that it works rapidly, but its effect is short-lived. Yes, it can work within about either 1 millisecond or up to about 10 milliseconds. Okay, so it can be rapid or it can be slow. Okay, that's what we see over here. So, this is the important thing that we have to understand regarding the electrical synapse and then the chemical synapse. Now, the first thing that I want you to know is that this chemical synapse, evolutionally, yes, it wasn't really supported. Why? Because it couldn't be compartmentalized. Whenever this one gets depolarized, this one gets depolarized. No. So we needed a well compartmentalized one, where there should be receptors to receive it and then decide what to do next. In any case, although we tend to find electrical synapse more in lower vertebrates, invertebrates, these kind of lower animals, and higher animals like vertebrates will be having the well compartmentalized form by way of the electrical synapse. What we see is that even in humans, the olfactory bulb, okay, very important in smelling, the retina, talk about the cerebral cortex, okay, talk about the hippocampus, okay, and importantly, talking about the lateral vestibular nucleus. We will be explaining all these things for you. They still show some kind of electrical synapse in there. But by and large, majority of synapses, you want to see the you want to see the chemical synapse. Okay? So, for now, that's where we will end this meeting. We just treated two things. In the subsequent videos, I'll be explaining, going more into some of these things for you so that you can really understand all these things. I'm very grateful for your time this evening. Have a good night, all of you, and may the good Lord richly bless you. Amen.